Hi, I'm John Carter, and this is our presentation on the article that we chose, which is entitled The Effects of Early Life Stress on Liver Transcriptomics and the Protective Role of EPA in a Mouse Model of Early Life Stress-Induced Adolescent Depression. Um, a quick introduction to this article is the article, The Effects of Early Life Stress of Liver Transnomics and the Protective Role of EPA in a Mouse Model of Early Life Stress-Induced Adolescent Depression, quite the title, describes research examining the relationships between early life stress, which we nicknamed ELS, the synthesis of eicosapentaenoic acid, EPA, in the liver, and the development of adolescent depression. The consequences of adolescent depression are far-reaching, and experience with various forms of abuse and neglect is strongly associated with an increased risk of depression in adolescents. Understanding the mechanisms in which early life stress may contribute to this helps us design intervention treatments, as well as therapeutic or medicinal products capable of reducing symptoms of this condition. The researchers in this study aim to answer the question, what mechanisms underlie ELS and its depressive impacts on adolescent liver metabolism, and does the supplementation of EPA provide relief to these effects? For this experiment, they used mouse pups, which are just young mice. And the two hypotheses that were posed in this study, their first hypothesis was that ELS would cause distinct alterations in transcription patterns of genes in the liver, particularly those associated with the biosynthesis of unsaturated fatty acids metabolism, which includes that of EPA. As a result of this, EPA levels in the liver would be reduced and contribute to the development of depression-like behaviors in the adolescent mouse pups. And their second hypothesis was that the supplementation of EPA to mouse pups may alleviate or reduce depressive behavior in mice exposed to ELS by counteracting the EPA reduction effects of the ELS of liver metabolism. And to do so, they used a range of methods to assess their hypothesis and invest the question. And we're going to go through all of the tests that they performed. All of these also included negative control elements to compare with the experimental unit, which was considered a normal population. The first part of their experiment was early life stress administration. So animal models, uh, the mouse pups, they were given a combination of maternal separation from days 7 and 15, and they were weaned early and put into a new cage for 6 hours a day. They were administered fluoxetine, which is antidepressant, and EPA, which is what we were testing in this research to assess the effects on depressive-like behaviors. To distribute these depressive and anxiety-related behaviors, they used an open field test, an elevated plus maze test, a sucrose preference test, a tail suspension test, and a forced swimming test. The anxiety-related tests were the OFT test, which was the open field test, which involved placing mice in a dimly lit room and recording their trajectory for 10 minutes. The elevated plus maze they recorded the trajectory for 6 minutes after placing the mice pups at the center of a cross elevation apparatus. And for the depressive behavior test, they used the TST test, the tail suspension test, which involved hanging the mice with tape 1 centimeter from the tail tip and 25 centimeters from the ground and recording the rest after 6 minutes. The forced swim test involved placing the mice in waters at 25 degrees Celsius and recording the duration after 4 and after 6 minutes, which sounds pretty unethical to us. We all agreed on that. The learning and behavior tests were used to stimulate, the effects of what might be depression in an adolescent mouse pup. A Y-maze experiment and the new object recognition experiment evaluated spatial working memory, and they were tested by placing the mice in a maze for 5 minutes and then testing the ability to recall the space a week later. Finally, to test the hypothesis of changes in genomic sequencing, RNA sequencing was used 24 hours after the behavioral test from liver tissues, and RNA sequences was performed. Biochemical assays were performed using aspartame aminotransminase and alanine aminotransminase, and they were measured in serum samples to assess the liver functions, and EPA levels were measured in blood samples. Staining was used to examine liver and brain tissues. Western blot and qPCR were used to analyze protein and gene expression. And finally, statistical analysis using a p-test and a t-test was tested on a significance level set at p.05. And moving on to the results of the experiment.
So my name is Emma Richards. I broke the results down into depressive-like behavior tests, anxiety-like behavior tests, and then liver and brain tissue damage between the control group and the ELS group. So for the SPT test, sucrose consumed by the rats decreased significantly in the ELS group. For the TST test, immobility time was significantly increased in the ELS group. And then in the FST test, immobility time was also significantly increased. These are for the depressive-like behavior tests. And then for the anxiety-like behavior test within the adolescent mice, between the OFT test and the EPM test, no significant changes were observed between the two groups. And then go to the next slide. That just shows the graph between the two groups for all of the tests that I mentioned previously. And then for the liver and brain tissue damage in adolescent mice, transcriptome sequencing was used. So in the KEGG pathway enrichment analysis, downgraded genes were observed and were significantly enriched in 30 pathways. The 5 most prominent pathways being steroid biosynthesis, protein processing in the ER, PPAR signaling pathway, cholesterol metabolism and biosynthesis of unsaturated fatty acids. And then HE staining revealed that no significant injuries within the liver and brain between ELS and control groups were present. And then in the EPA content, that revealed that significant content reductions were measured in the liver and hippocampus of the ELS group. And then these graphs just show the statistical data of the liver and brain damage between the two groups. And then if you go to the next slide, that shows actual pictures of the liver and brain damage between the two groups.
So my name is Elizabeth Mormon, and I just kind of went to the interpretation of the results a little bit. So the first interpretation of the results in this study is that ELS induces depression-like behavior in adolescent mice. And this was shown by the results from the depression-like behavior test, which is why I agree with the authors in this interpretation. Specifically that there is a decrease in sucrose consumed and increased immobility time observed in the tail suspension test and forced swimming tests in ELS exposed mice compared to the control group. So this all indicates that ELS exposed mice had depression. A second interpretation of the results is that ELS causes a significant reduction in the levels of EPA in the liver, serum, and hippocampus, and this was shown in the EPA assay. Additionally, plasma levels of EPA are decreased in patients with adolescent depression. So all of these results from this specific part of the experiment leads me to agree with the authors in the interpretation that depression causes a significant reduction in the levels of EPA. A third interpretation of the results is that ELS induces abnormal biosynthesis of unsaturated fatty acids metabolism in the liver of adolescent mice. I agree with the authors in this interpretation because the Western blot showed a significant separation between the control and ELS groups, specifically altered transcriptional patterns in the liver of ELS-exposed adolescent mice. Particularly, there is shown to be a decrease in the expression of rate-limiting enzymes involved in the biosynthesis of EPA in the liver, specifically fatty acid desaturases 1 and 2. Additionally, fatty acid desaturases 1 and 2 have been shown to play an important role in neuropsychiatric disorders such as depression. So all of these results lead me to agree with the author's interpretation that depression and reduced EPA levels are related. Another point of support with the author's interpretation is that EPA content in the brain is lower in several psychiatric disorders. A final interpretation to kind of summarize all the results is that supplementation with EPA may be beneficial to the treatment of adolescent depression. And because the correlation between decreased EPA and adolescent depression has been shown, it makes sense that increasing EPA levels may be able to treat depression. Additionally, the experimenters proved that EPA administration resulted in an increase in the percentage of sucrose consumed and decreased immobility time during the tail suspension test and forced swimming test, all of which are indicators that the ELS mice performed better in the depression-like behavior test compared to before treatment with EPA. All of these results together lead to the interpretation that increasing EPA levels mimics the effect of antidepressants and therefore can be used as a treatment for depression in adolescents.
My name is Melanie Nunez. In conclusion, early life stress was found to increase the levels of adolescent depression in mice. Research also shows that changes in the transcription pattern in the liver are also associated with this. The study found that early life stress led to depression-like behavior in adolescent mice and altered gene expression related to the metabolism of unsaturated fatty acids in the liver. According to the article, early life stress reduced the levels of EPA in the liver, serum, and hippocampus as well. Administering EPA showed signs of improving the depression-like behavior that was caused by early life stress. Therefore, the study suggests that for adolescent depression that is associated with early life stress, EPA can be a potential treatment to help bring depression levels down. Some of the future ideas for this study, with continuing this research, this topic could help make newer and possibly more efficient treatments using higher levels of EPA to bring early life stress levels down. An example of a future idea is researching whether a change in diet by eating EPA-rich foods can change a person's depression level. There are many factors to look at in this study, such as genetic predisposition of EPA levels, but plenty of progress is being made.
Here's the link to the article. Thank you for watching our presentation.